On October 20th, 2016, a Cell paper brought a new scientific step toward uncovering the genetic coding which affects the loss of limbs in certain vertebrates. Astounding research has been conducted by Dr. Alex Weiss, a geneticist at the Lawrence Berkeley National Laboratory. He and his colleagues expressed interest in determining the genetic factors that lead to the loss of snake limbs. Through genetic genome editing methods, they found key information in the snake DNA that leads scientists down the right path to discovering the answer. Through the study of snakes, scientists have found they lost their limbs over 100 million years ago, but they have not found the genetic reasoning behind it.
Remnants of bones known as vestigial limbs are still present in many snakes today, and because of this, snakes are considered tetrapods.
A tetrapod is an animal that has 4 legs. This includes amphibians, mammals, and reptiles.
It's very difficult to find snake remains due to how easily their tiny bones break down, but luckily, evidence has been discovered. In 2006, a fossilized snake was found during the mid-Cretaceous period. This snake had fully functional hind limbs. Also, in 2007, a snake dated back 95 million years ago was found with fully functional hind limbs and vestigial front limbs.
For these reasons, although snakes don't have 4 functional limbs, through their ancestors' traits, they are classified as tetrapods.
Snakes lost their limbs due to genetic mutation called deletion. Deletion is a part of a chromosome or a sequence of DNA that is lost during DNA replication. Fine.
The fact that snake lost the ability to have legs is very odd. Deleterious mutation dominance only occurs 0.2% of the time. Snake classification consists of Kingdom Animalia, Phylum Chordata, Class Reptilia, Order Squamata, and Suborder Serpents. Snakes break off from other lizards in the subgroup Serpents.
The sonic hedgehog gene, or SHH for short, is a gene involved in limb formation. Diving further into SHH, you will find a stretch of DNA called the ZRS.
In snakes, the ZRS is missing 17 base pairs. Another factor that prohibits snakes from growing legs is the transcription factor binding sites. These affect the way gene info is transcribed. In snakes, the 3 activators of the sonic hedgehog gene switch are deleted, and therefore the SHH turns on and then permanently off during the limb growth process.
A combination of these 2 factors resulted in snakes being incapable of having limbs. This had sparked interest in researchers from Lawrence Berkeley National Lab, specifically Dr. Alex Wissel.
Due to his interest in the matter, Dr. Alex Wissel compiled a group of scientists to study it. They took snake ZRS and inserted it into the DNA of embryonic mice using advanced genetic technology called CRISPRs. And the result? Mice with no limbs.
As shown, mice have fully developed appendages, and cobras and pythons both have truncated limbs. One difference demonstrated here between the 2 snakes is the sizes of the vestigial extremities. Pythons are more primitive, having larger limbs than cobras. This shows different stages of the evolutionary process, even in the same subgroup.
Even though the truncated bones serve no purpose in movement for snakes, there are a few things they are used for. In primitive snakes, the remnant bones are used for courtship and sexual combat. However, because this happens in so few snakes, the limbs are still considered vestigial.